Good morning, esteemed judges, fellow researchers, and distinguished participants. My name is Kirana, and I'm honored to present my research on the effect of lycopene administration on the number of neurons in the cerebrum and cerebellum of mice exposed to lead acetate. This research is part of my academic work at the Faculty of Veterinary Medicine, Universitas Airlangga. Here is a bit on the outline of what I'm going to present today. Lead is still one of the most persistent environmental pollutants worldwide. Its presence in industrial waste and fossil fuel emissions allows it to accumulate in the ecosystem, eventually entering the human and animal body through the food chain. What makes lead especially dangerous is its ability to cross the blood-brain barrier, where it triggers oxidative stress and damages neurons, particularly in the cerebrum and cerebellum. On the other hand, lycopene is a natural antioxidant found in tomatoes and other red fruits, and has shown promise in reducing oxidative damage and inflammation. So the question is, honorable participants, can lycopene actually protect brain tissue from lead-induced damage? To answer that, the study aimed to evaluate the effect of lycopene on neuron survival in two critical brain regions, the pyramidal neurons of the cerebrum and the Purkinje cells of the cerebellum. We proposed that lycopene would have a neuroprotective effect. We designed a controlled laboratory experiment with 25 healthy female mice, using a completely randomized design. The animals were randomly divided into 5 groups. One group received distilled water, another received only lead acetate, and the remaining 3 groups were treated with lycopene doses of 5, 10, and 20 milligrams per kilogram body weight, respectively, following lead exposure. All treatments were given orally via gavage for 14 days, and on the 15th day, the mice were euthanized, and their brains were extracted and prepared for histological analysis using hematoxylin and eosin staining. We then quantified the neurons using ImageJ software, and the data were analyzed statistically using ANOVA with a significance level of P under 0.05. Here you can see a graph of the order of treatments I explained before. This study was ethically approved by Universitas Airlangga's Ethics Committee under the certificate number as stated. Our results also showed what we had expected and hoped to see. In the positive control group, which received lead acetate without lycopene, there was a clear drop in neuron numbers in both brain regions compared to the untreated group. But with lycopene treatment, especially at the highest dose, neuron counts improved significantly. For example, in the cerebrum, neuron counts in the lead-only group averaged around 568, whereas the group that received 20 milligram per kilogram body weight of lycopene, the count increased to 721, nearly reaching the healthy control group's level. Here you can see the histopathological slides from each group, which shows the difference of neuronal count. In the cerebellum, the neuron count dropped to 60 in the K plus group, but improved to 88.4 in the P3 group, again approaching the control value of 92.4. Here you can see another part of histological slides in the cerebellum. So, what does this tell us? The findings support the idea that lycopene helps protect neurons and its effects increases with dosage. This can be explained by lycopene's ability to neutralize reactive oxygen species, enhance antioxidant enzyme activity, and reduce inflammation at the cellular level. It's also important to note that lycopene's lipophilic nature allows it to integrate into neuronal membranes, where it can stabilize mitochondria and prevent apoptosis. These findings demonstrate that lycopene provides dose-dependent neuroprotection. This is attributed to its ability to neutralize ROS, prevent lipid peroxidation, activate antioxidant pathways, stabilize mitochondria, suppress inflammation, and enhance the resilience of cerebral neurons. However, the results are promising. Our study also had limitations. We focused only on short-term exposure, and we did not assess behavioral or functional outcomes. We also only used female mice. These are important areas for future research, particularly if we want to evaluate the real-world therapeutic potential of lycopene. To summarize, lycopene showed a significant and dose-dependent neuroprotective effect in mice exposed to lead acetate. At 20 milligrams per kilogram body weight, it was especially effective in preserving neurons in both the cerebrum and cerebellum. These findings suggest that lycopene may have value not just as a dietary antioxidant, but also as a potential therapeutic agent in managing neurotoxic exposure in veterinary and medical contexts. For future studies, I recommend exploring long-term effects, incorporating behavioral testing, and examining molecular mechanisms more deeply. I would sincerely thank the Faculty of Veterinary Medicine, Universitas Airlangga, especially Professor Dr. Wijiati for her supervision throughout this study. Also, thank you to the laboratory teams who supported the experimental process. This research was conducted under ethical clearance with the number as stated. Thank you very much for your attention. I welcome any questions you may have.